Morning, everybody. Mrs. Rayberg here with notes on 4-4, all about different forms of photosynthesis and things that affect the rate of photosynthesis. So we're going to start out here. So this is 4-4, alternative forms of carbon fixation, which is just kind of another term for photosynthesis. So we talked the other day about how leaves on the underside of them have these little holes in them where the gas exchange occurs. And for most days out of the year, it's perfectly fine to open those up and let CO2 go in and O2 come out. But in the extreme temperatures of the summer or in the really cold, we don't want to keep those little holes open all the time because we get a lot of water loss. So on really hot days, say it's like 100 degrees outside, the water that's in the plant can also escape out as water vapor, and that can cause the plant to lose water pressure and slow down the rate of photosynthesis. So we don't want that to happen. So on really hot days, plants will, in the middle of the day, close their stomata, remember, which is just plural of stoma, which just means one hole, in order to prevent the water loss. But it also prevents them from absorbing carbon dioxide. And if they can't absorb carbon dioxide, they can't do the dark reaction. So they got to figure out a way to deal with this. So here is the norm. This is what most plants do. That type of photosynthesis we refer to as C3 photosynthesis, or we call them C3 plants. The reason why we call them C3 plants is because, as you remember, you have the carbon dioxide that comes in and joins with the 5-carbon RUBP, and what that does is it turns into the 2 3-carbon molecules called PGA. So we call this a C3 plant because of the 3 carbons that are made initially during the dark reaction. So we call this a typical form of photosynthesis because it makes the 3-carbon PGA during the process of the dark reaction. So some examples of these plants would be rice, wheat, and soybeans, although the majority of plants that we see outside would be considered a C3 plant. Okay, the next type we have are called C4 plants. There goes a really loud airplane. C4 plants because as they're doing photosynthesis, when the carbon dioxide comes in, instead of making a 3-carbon molecule, it makes a 4-carbon molecule called PEP, which stands for phosphoenol pyruvate. There's the P, there's the E, there's the next P, so we'll just call it PEP. And so they make this 4-carbon molecule when photosynthesizing. This produces a greater rate of glucose per carbon coming in, and so they tend to be more efficient when the temperatures are hot and the stomata are partially closed. So these are things like your front yard, grasses, sugarcane, and corn. A lot of the, I don't know if you remember this word, monocots, the leaves that have parallel veins like this, as opposed to leaves where it branches out like that, which are dicots. So if it's got a parallel type leaf, it's probably a C4 plant that makes, PEP when it's doing the dark reaction. And so what happens in these is that the stomata, instead of being completely open during the day, they're only partially open during the day. It's kind of hard to tell there. So they are partially open, partially closed during the day, especially when it's a little warmer. And then we have plants that have to deal with really, really hot temperatures, and we call these CAM plants. CAM plants are the things that we usually find in really dry, arid means dry, hot temperatures. Desert. This is very much a desert way of photosynthesizing. So what they do is during the day, they keep them closed. So during the day, the stomata are completely closed. No gases going in and out. But at nighttime, when the temperatures are cool, they will open up the stomata and allow all sorts of gases, CO2 especially, to go in. All their oxygen pops out. And then, because they're not going to lose any water, it's nighttime, it's cooler most of the time, and so they don't lose as much water vapor that way. Because think about it, if a desert plant were to open its stomata during the daytime, they would be losing so much water they would shrivel up and die. So if they just suck up as much CO2 as they can during the nighttime and then close it off and then use it for the dark reaction during the daytime, that actually prevents them from water loss. So we call these CAM plants, and I can't remember exactly I can't remember the name of it, but it doesn't matter, you don't need to know it anyway. But just remember, these are juicy plants called succulents and cacti. Is what you'll find these guys. And so they just keep their stomata closed during the daytime, open them at night, suck up CO2, and then continue during the day. Now, we've been messing with in the lab different ways that I can affect the rate of photosynthesis. There's three main things that affect the rate of photosynthesis, three factors that affect it. The main one being, or one of them being, carbon dioxide concentration. So that was the first experiment we did with messing with the concentrations of carbon dioxide in the water by adding different amounts of baking soda. So here's what happens. When the CO2 concentrations are low, then the rate of photosynthesis is really low. Because remember, CO2 is one of the main ingredients of the dark reaction. So if I don't have a lot of those, I can't make a lot of sugar. But as the CO2 concentration increases, so does the rate, until it finally hits a peak. Why does it hit a peak and flatten out? Well, it's because the dark reaction could only go so fast. So I can inundate you with a plethora of carbon dioxide, but it doesn't matter. If I can only go so fast, I could have all the carbon dioxide in the world, but if I can't go any faster, my rate flattens out. So photosynthesis will increase as CO2 increases, then it hits a peak, and then it's done. Okay? The next one is light intensity, and this has to do with how bright it is outside, but also has to do with the color of light as well. So we said that, I'll draw a little line here, if this is the purples and these are the reds over here, we know it really likes the purples and the blues, doesn't like green, but it kind of likes red. So green's right there. So in here, over in the purple side, is high energy, and over here is lower energy. And so we can see that they like these colors the best, and that light intensity. So same kind of idea. Light provides the electrons needed for the electron transport chain to work. And so if the light is low or dark, we have no photosynthesis. As we make it brighter, it increases and increases until, again, it flattens out. Why? Because same reason for CO2. I can inundate it with as much light as you want, but once it hits its max speed, it can't go any faster. Well, this right here is the max speed. It can't go any faster than that. So it doesn't matter how much light I give you, I'm only going to be able to go so fast. Alright, and then the last thing is temperature. So here's cold, warm, and really hot. So photosynthesis has a temperature that it likes to work at, and it depends on the plant too and where it grows. This could be 80 degrees, it could be 60 degrees, it could be 100 degrees. But there does come a point when once you start getting warmer, then photosynthesis begins to slow down. Same thing when it's colder, it begins to slow down. Why? Because the hotter the temperatures get, a lot of the reactions going on are caused by enzymes. And what do we know about enzymes in warm temperatures is that the warmer an enzyme gets, the more likely it is to denature. And when it denatures, that means it falls apart and it doesn't work anymore. And so once I get beyond a certain temperature, I've completely denatured the enzymes and it doesn't work anymore. And so therefore photosynthesis won't occur. Same thing, if it's just too cold, my enzymes aren't working. So there is an optimal temperature that they enjoy the best. They will work in a range of other temperatures. Not as fast over here, not as fast over here. So that's another experiment we're going to do, is test the temperatures of the water to see if that affects how much it goes to. Alright, so that's it. That's actually everything for this unit. So go back and review your notes. Here come the children. Go back and review your notes. Look at Study Blue. Start getting all those vocab terms in your brain, and then we'll continue with the lab in checking out how rates are affected by all these different factors. Okay, so we'll see you later. Bye.